From Microbe TV, this is TWiP, This Week in Parasitism, episode 119, recorded on October 25th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month, and for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. This episode is sponsored by Drobo, a family of safe, expandable, yet simple-to-use storage arrays. Drobos are designed to protect your important data forever. Visit drobo.com to learn more. Hi, everybody. I'm Vincent Racaniello, and joining me today, right here in New York City, Daniel Griffin.
Hello.
And that's it. You and me, right?
Just us. No Dixon today.
Dixon's in Bolivia.
I know. He's every so often I get an email from Bolivia. It's a very short email, so I don't know what his internet access is. But what I did is, people often, when they hear an episode without Dixon, they miss him. So I'm going to try to bring a little bit of Dixon. Like, for instance, I printed out all the emails, even though I have a computer in front of me.
Wonderful. You printed out all the emails. That's great. What else? Let's see. What else does he do? He doesn't fall asleep. I know that.
Well, also the humor. People always like his humor. Sometimes I've actually had people tell us that you and I are too serious.
We are.
So we'll
have
to introduce some levity here.
Oh, too serious. I don't know. I thought giving him a hard time was pretty funny.
I think it is. So we'll have to make sure, even in his absence, we bring him up and poke fun at him.
That's funny. Too serious. Wow. Well, that's what he's here for, right?
Exactly. He's a man who enjoys life.
Well, he sure does. He's enjoying it. He's enjoying it in Bolivia.
Right. You and I are working, and he's off in Bolivia. So
I think he's coming back Friday, right?
I do not know.
Well, this is the first episode you and I have done together. We had one without him, but Paul Cali was here.
That's true. It's true.
But you and I, and I guess this is going to be a very serious episode.
Very serious.
What can we do to make it funny and levy? Well, it's a nice day out, right?
It is.
I just saw this plane fly by the way, where the motors, the props were like pointed upwards.
So Martine, who's the administrative assistant to Steve Goff, was actually, I was, unbeknownst to you, I was in your office watching it while you weren't here and pillaging through all your stuff. No.
There's nothing to
but no, apparently these, what do they call them, hawkers or something? There's I forget their name, but those are those sort of a fixed wing aircraft, but has the ability to rotate the propellers and take off vertically. And apparently they're quite popular among the higher up in the military. So whenever Congress realizes that we're spending, I think, a quarter of our yearly budget on these, they try to quash them, but the higher ups say, no, we need them. They're so cool.
So you can take off vertically.
Exactly, from anywhere.
And then they tilt the props a little bit.
And then the props tilt, forward, and they actually travel like a fixed wing aircraft.
I've never seen. I know when I was younger, I remember they were experimental, but I didn't realize that they actually make them. So we flew right by. I guess they're flying around several times because I saw it, and then you saw it when I wasn't here.
Yeah, what we should say, fixed wing vertical takeoff. Because I was sort of thinking, so the VTOL, right? That's
VTOL.
Helpful.
The one I saw was a propeller plane, not a jet.
Exactly. Yeah, I've never seen ones that are jets, but actually, I guess I'm seeing here
the Harrier is.
I think that's what we were seeing as Harriers. But are you
I saw
the
propellers.
Osprey?
I saw propellers. Oh, Osprey.
So maybe it was the Osprey.
Is that what it's called? VTOL,
which are somewhat newer. I think they came out in the end of the 80s.
It's an Osprey.
Yeah.
For sure. Bell Boeing V-22 Osprey. Now, there it is. That's exactly what flew by. Yeah. American multi-mission aircraft with vertical takeoff and landing, and also can go forward. which is, of course, what you want to do.
Exactly.
So there's doing that in the helicopter.
Yeah, I think that they are faster. Is that what it is? That the Osprey is faster than the, I don't know.
Both vertical takeoff and landing. So that's a helicopter.
Yeah.
It combines the functionality of a helicopter with the long-range cruise performance of a turboprop aircraft. Okay, so you get longer range. You probably get more room also, right? I don't know.
Probably. They look pretty big, but I guess now we could have a horizontal takeoff of our show. We can start.
Yeah, let's do that. We had a case for last time. Actually, it was more than 2 weeks since our last case, right? Episode.
It actually was a little bit of time. We were gonna, and I think that's why we had to jump ahead because
7th, yeah.
With Dixon being gone, it was 3 weeks.
Yeah, and we needed to get an episode in for October. We weren't going to do a second episode. Need to do 2 per month.
Yeah,
right. So here we are. We had a few guesses, but why don't you remind us what we're doing here?
Yeah, this was a little, as I said, a little bit of a twist. This time we had a family. A human family, I clarified with that. It was a human family with eosinophilia.
Right.
And as I mentioned, these series of cases were initially conveyed to me by my good friend, colleague, and ID physician down in Australia. She actually published this series of cases. Elaine Chiang, shout out to a really bright ID doc in Australia. And the story is we had a 45-year-old man. He was having problems with mild abdominal distension seen by a doc in Sydney, and a CBC showed eosinophil count of 10,500. That's not his weight. That's the eosinophil count, so quite high. His wife of the same age reports feeling fine, but when she gets a complete blood count, she also has eosinophils that are elevated. Hers are above 5,000. They have 2 daughters, one 17, one 19, both without symptoms. One, however, has some elevation in eosinophils, 900, where normal's up to about 500. The other one, also no symptoms, has no elevation in eosinophils. 1 week prior, we get the story, 1 week before the dad starts having symptoms, the sister-in-law came and visited them. She's also from the same area of Australia, and she stayed with them. And while she was there, she developed severe diarrhea, abdominal discomfort, bloating. She started losing weight, which continued. Her eosinophils were just under 5,000. And when we go through the history, we do find out that they do make a habit of eating raw fish that they buy in the local markets. We don't find out whether it's fresh or saltwater fish. No overseas travel. They don't leave this somewhat urban, semi-urban environment. No pets, no homegrown foods. And we went back through previous labs. We found that the labs had all been normal without an elevation in eosinophils prior to this. So this is new. And with a full workup for a bunch of different things, we find out strongyloides is negative. HIV is negative. We don't have any toxic habits. And I think that was where we left things.
Now, as far as I could tell, the fish is the key here, but maybe it's a red herring.
I think we had some people email making that comment.
Because I thought about this in terms of a fish-acquired infection, and but maybe we'll find out that it's not. We had,
I mean, I guess when we're done with all the emails, we'll discuss a little bit about probably eosinophilia, what causes it, what to think about. But
all right, we had 4 guesses. The first is from Eric. Hello, TWiP crew. This is my first time writing in with a guess for the case of the week. I felt compelled to write in because it turned out that my guess for last episode's case was correct, and on hearing this week's case, I felt that I had a pretty strong guess without even having to utilize Professor Google. The staggeringly high eosinophil count in the patients immediately made me think that this is likely an infection with some parasitic worm or other. The symptoms imply that the infection is in the GI tract, and while I admit the mention of raw fish consumption could be a red herring, pun intended, the symptoms associated with the parasite that I'm guessing are too close a match for me to think it's anything else. I think the patients have a case of anisakaiasis. After double-checking on cdc.gov, anisakaiasis symptoms include abdominal pain, nausea, abdominal distension, and diarrhea. Unfortunately, I can't guess any particular species because I don't know enough about this parasite. I don't think I have any good differential diagnosis either. Hope I got it right on my first time writing in. Maybe I'll submit guesses more often.
Do you think he's only going to write in if he's correct?
You never know. But I have to tell you, Eric made us these books, Dixon and I.
Oh, those are very nice.
He's a book binder, right? And he bound this himself.
Oh, wow.
And he got influenced by TWiP. He said it made him want to pursue virology as a career, so he's grateful, and he gave us these. And Dixon's book has a nurse cell on the cover.
Oh, wow. I mean, those are really neat. Yours nice. Has the... I'm assuming that's a virus?
Yeah, little virus, an icosahedral. Yeah. Okay. So he's an anisakiasis. Okay, let's see.
Wink writes, Dear TWiP professors, my guess this week is gnathostomiasis. I think it can be found in Australia on freshwater fish. For the first few weeks of infection, you get GI symptoms. Later, you may get skin swellings and errant parasite distribution. Wink Weinberg.
Mm. All right.
Maybe we'll have to do a case of gnathostomiasis. I'm letting you know ahead of time, Wink. It may not be that, but I actually think that's a good guess.
Mm. Jeff writes, Dear TWiP doctors, it's sunny 65F in South San Francisco today. An interesting case, as most of the family was infected. When thinking about parasitic infections from fish, usually you think of Anisakis simplex for saltwater or Diphilibothrium latum for freshwater fish. The clinical presentation isn't consistent with anisakiasis, so I would rule that out based on the presentation. Diphilibothriasis is more consistent clinically, but there are a few things which make this also unlikely. A number of cases of Diphilibothriasis are asymptomatic, and most of the family members have disease. Furthermore, Daniel didn't mention the presence of anemia in any of the family members, which is common in cases of Diphilibothriasis and resembles pernicious anemia. So I am really going out on a limb here and guess Giardia duodenalis as the infective agent, as it is consistent with the clinical presentation, and I have found some evidence of this being important in the region. I ran across this reference in my research and thought it was pretty interesting. International Journal for Parasitology, 2010. Well, there you go. Jeff has been often right.
Yeah. Michelle writes, this is a long one. And she's a long-time listener. I am a long-time listener, but first-time writer. I am an assistant professor of microbiology at a medical school in the Pacific Northwest. Although parasites are my specialty, especially schistosomes, I do not get to lecture about them very often. Unfortunately, our curriculum is limited when it comes to eukaryotic pathogens. This is where Dixon would moan or something about the loss of this education.
Right.
Which is actually really, it's unfortunate, but we'll get to that. I love your idea of giving out your textbook to all medical students. The trick is getting them to actually read it. Students these days appear to be afraid of textbooks. This is true. Actually, no, we're gonna do that on Halloween. I don't know if you know. You're welcome to join us, but Dix and I are going to walk around the neighborhood with textbooks to frighten the children.
You'll scare them.
Okay, for the family.
Not because of the pictures, just the textbook.
No, just the fact that it's a textbook. For the family in Australia, eosinophilia plus a range of symptoms from asymptomatic to abdominal distension to severe diarrhea and cramping suggests an intestinal helminth. The clue of raw fish connecting the cases leads to the hypothesis of infection with the broad fish tapeworm or Diphilibothriasis. Where's Dixon to correct my pronunciation when I need him? Although this parasite is typically thought of as having a circumpolar distribution, further research on Google Scholar indicates that the distribution of this disease appears to be expanding. In fact, it is included in the CDC's Emerging Infectious Diseases Journal. And she actually gives us Kutcha et al. 2015. A paper from 2009, Schultz et al. 2009, reports no reliable recent reports in Australia. However, there is a recent case report from Australia of a 3-year-old infected with the tapeworm Adenocephalus pacificus. Infection was presumably acquired from eating raw marine fish caught by his father off the southern coast of Australia. The tapeworm, Adenocephalus pacificus, was known to be endemic to Australian territories within pinnipeds for quite some time, which suggests that recent infections have more to do with changing dietary habits rather than imported parasites, although imported cases have been documented in other areas from South America to Spain. There are several species of Diphilibothrium and Adenocephalus, and their life cycles vary somewhat, but the general pattern involves 3 hosts: a copepod, those are the water fleas, an intermediate host, a fish intermediate host, and a fish-eating mammal definitive host. They do not seem to be very specific to their mammal or fish hosts. The tapeworms are reproductive adults within the intestine of fish-eating mammals. They release eggs within the host species into the water, and when the eggs are fully developed, they hatch, releasing a ciliated coracidium. The coracidium is eaten by a copepod, a small crustacean, and develops into a procercoid within the body cavity. When an appropriate fish host eats the infected copepod, the parasite develops into a plerocercoid. Several marine, brackish, and freshwater fish species can serve as hosts for the different species of these tapeworms, with salmonids being a common source of human infection. When the mammal eats the plerocercoid in the fish musculature, the tapeworm establishes in the intestine and matures into an adult. The symptoms caused by infection with these worms are variable and depends on the number of tapeworms present. The individual's response to infection and probably characteristic of the tapeworms, although this is not well understood. Many infections are asymptomatic, and symptomatic ones include diarrhea, abdominal discomfort or pain, nausea, vomiting, and bloating. Diphilibothriasis is also classically associated with vitamin B12 deficiency and thus megaloblastic anemia, although this presentation is apparently rare and may not be caused by all species. Kutcha et al., 2015. For diagnosis, sedimentation of feces should reveal characteristic eggs. The eggs do not float well when zinc sulfate or a sugar solution is used to concentrate the eggs, thus sedimentation techniques are necessary. Egg shape varies somewhat among the species, but they are about 40 by 60 micrometers, oval to round in shape, operculate, and have a knob or small protrusion at the abopercular end. Chains of the proglottids may also be found in a macroscopic examination of fecal samples. The proglottids are more wide than long, hence the name broadfish tapeworm. Identification of the species can be accomplished through DNA sequencing. Praziquantel or niclosamide can be used for treatment. Keep the cases coming from the ultra-rainy, even for us, Pacific Northwest, Michelle. And then we've got some references here. And she is an assistant professor of microbiology at, it says Comp NW, Western University of Health Sciences, Lebanon, Oregon.
Lebanon, Oregon.
Wow.
Oregon
is what we say.
Say Oregon.
Pronounce it properly. That was quite the email.
Oregon. Let me just see where this is in reference to the population of 15,000. Small. Let's see. I'm zooming out on the map here.
Okay.
It is south of Portland, kind of due south of Portland, about as far inland as Portland is. Okay, now I know. So it's like halfway north-south in the state. Well, here's my logic was, I figured the fish were involved, and I went to... it's a good Wikipedia page on anisakis. They have a nice picture, symptoms of raw fish infection. They have 3 distinct symptomatologies. Clonarchiasis, where you would get jaundice, liver enlargement, GI symptoms, right? Then anisakiasis, sudden severe pain.
Yes.
And I didn't think that fit with this family.
Yeah, that was sort of the tough thing is that, and it is, I think we had a case, right? The Japanese student, remember? And she made some homemade sushi and it was a boyfriend involved. And it was severe enough that, it prompted an ER visit.
Because the worm had come out of the fish and was trying to burrow into her stomach.
Exactly. Right? It's actually burrowing through.
Not long after she ate the fish. And then Diphilibothrium, where you have some irritability and tingling of the skin, but most of the symptoms are abdominal, vague abdominal discomfort. I kind of thought that went more in line with our family, especially since a couple of them didn't have any symptoms. So I was going with Diphilibothrium because also you get eosinophilia. It's fish acquired, although the problem, as Michelle pointed out, it's not associated with, none of these are really associated with Australia. That's the problem.
Yeah.
Anisakiasis is not really down there. It's elsewhere. So
it's true. And the Adenocephalus, which is the other broadfish tapeworm, is, if you go to CDC and you look at the distribution, you're going to be seeing a lot of Peru, a lot of West Coast South America. Again, not a major player in Australia.
Yep. And these people hadn't traveled, right? They'd stay home.
They're hanging out
eating
there.
Their fish. So let me the fish, that came from a question that they were asked, right?
Yeah, that somehow, and Elaine writes this up, somehow they seem to pin that down, and we're going to suggest as we go forward that may be related to how they acquired this infection. So I do think there's a few things that we can, it is tough. A lot of times people go backwards with the history. Once they know the diagnosis, they start asking questions, right? If someone has echinococcus, next thing you know, tell me about sheep and dogs. Right. Or I had a case where I think I was in India and everybody was thinking brucellosis.
Yeah.
It actually, interesting enough, was a milioid infection of the sacroiliac joint. But it was this IT worker, and when I went to talk to him, there was some other people in training asking him questions, and he was like, "Oh yes, the goat milk. I drink it right from the goat."
Oh my god!
And I was like, "Stop giving them a hard time."
The goat milk.
But yeah, so you ask the questions sometimes when you already know the diagnosis, and I think once they were considering the diagnosis, they went back and went. So the way I started thinking about this case when my colleague was presenting it was, okay, the fish exposure, which maybe is an issue. And I have to admit, at the time, I did not know the association between the fish and this pathogen. And the other was the eosinophilia. And we can narrow ourselves down to parasites. We don't, we're not going to be, I'm kind enough not to be presenting a connective tissue disease or a neoplastic or allergic or just an odd idiopathic manifestation. So we've got a parasite, but then the next question is which parasites cause eosinophilia? And classically, tissue invasive helminths would be a group that causes eosinophilia. So let's say you have ascaris worms in your stomach. Maybe you've got a ton of them. You're not going to necessarily have eosinophilia then. You're going to have the eosinophilia as the ascaris is going through its migration phase before it ends up back in your stomach. So you end up with the eggs in your stomach, and then the juveniles wander all over the place. They go through your lungs. You get eosinophilia. And then it's this tissue invasive stage when you have the eosinophilia. Once they get to where they want to be, back into the stomach, then the eosinophils go away. But that is your class. And that's actually tough as we notice, because let's say it was a tissue invasive helminth. It's hard to diagnose them then because they're not adults. They're not making eggs yet. Often the diagnosis is after the fact. Now, schistosomiasis, I think our colleague, is a tissue invasive parasite, and it's going to have a similar sort of process where you have this Katayama fever, and you don't even have the adults yet. So we think about tissue invasive stages of certain of parasites. The other, and I think it's an exception, is we can think about certain protozoans. And there are certain protozoans that can give you eosinophilia. And the classic ones we talk about are Sarcocystis and Cytoisospora belli. That's kind of the one that everyone remembers. Dientomoeba fragilis, which is an amoebic protozoan that can actually give you diarrhea, abdominal discomfort, and prolonged eosinophilia. And then, as I think people are mentioning here, occasionally you can actually, even though you think of these broadfish tapeworms as intestinal, there have been reports of eosinophilia with them. So they're actually, I have to say, excellent thoughts, and something, until we get the results we're going to get, I think they would be excellent things. And they were things that I had on my list. And when I was having this case presented to me, I was a little bit cynical at the time. I'm not sure I buy this. It took me actually a while until I think the treatment. So let me maybe talk about what we find next. I will pull it out.
So you heard this in a presentation initially, You didn't know what it was ahead of time.
I didn't know what
right?
it was ahead of time.
It was a formal presentation on rounds or something. Right?
So I was down in Peru, and Elaine was, presenting this case to a number of us.
And then, does she go right to the solution? Does she wait and hear?
No, she lets us embarrass ourselves.
Did you guess correctly?
No.
What did you guess?
I think I may have actually been thinking of a fish tapeworm at the time myself, in all honesty. Mm-hmm. And then even when she presented it, it actually took me a little while before I read the literature and was convinced that she was correct.
Interesting.
Because she was actually in the group that was first describing this as something that existed. So as I mentioned, they did a lot of these tests, but now this is they do stool testing, and I'm going to give you the stool. I'm going to give everybody, all of us, the stool test. So the first person they test is the father, and they see several things in his stool. They I say evidence of several things. So, sometimes they're seeing. Well, okay. So the father had Blastocystis hominis, Endolimax nana, Entamoeba hartmanni, and high numbers of Dientamoeba fragilis in his stool. Okay, so that's the dad. The wife, again, who has the high eosinophils, she has Blastocystis hominis, Endolimax nana, and, again, high levels of Dientamoeba fragilis. The two daughters, right? One has high eosinophils, the other doesn't. The one with elevated eosinophils only had Dientamoeba fragilis in her stool. The one without high eosinophils, nothing was detected. Even they did 6 stool studies. They really were trying to find something. And the visiting sister-in-law, she had Endolimax nana and high numbers of Dientamoeba fragilis. Okay, so they go ahead. They actually do PCR. At this time, the idea is that maybe the common thing that they're seeing in everybody, the common thing is the Dientamoeba fragilis, because all of them had this except for the asymptomatic daughter. And so they go ahead and they treat everybody except for the one asymptomatic, no eosinophil daughter with Flagyl, with metronidazole, 3 times a day for a week. And the eosinophil counts drop. There's complete resolution of symptoms in the dad and the sister-in-law. But the daughter and the wife continue to have high eosinophils. So now they treat them with doxycycline. Because when they did repeat studies, they still saw the Dientamoeba fragilis in their stool. So now they treat them with doxycycline, and then they go ahead and treat them with paromomycin. And now the stool clears. They don't see any more of the Dientamoeba fragilis. And they do 12 months later, everybody's symptom-free. No one has the eosinophils. They check the stool, and the stool is clear. So that was what she presented. And now, actually, at the time, right, this was a little while back. And so, there hadn't really been a sort of a full literature, but now there's actually been several family clusters and cases, similar presentation, the eosinophilia, they isolate the Dientamoeba fragilis, they treat, and actually you get resolution of symptoms, clearance of this protozoa, this amoeba from the stool, and the eosinophils.
Look at this. I just searched for family clusters of eosinophilia, and bingo, Dientamoeba fragilis.
Comes right up.
I mean, I didn't think of it that way because I only thought of this one isolated family. Dientamoeba has emerged as an important and under-recognized cause of GI illness. We report a familiar cluster. I wonder where this cluster was. Let me look. This is 2013.
Okay.
Let's see if they tell us where it is. And there have been others, I guess. Oh, my gosh.
Is it Australia?
What was the young lady's name?
Is it Elaine Chiang?
Yeah, she's on the paper.
Okay, so she's on this one.
That's
too.
her paper. Is it this family?
No, I think there was another one, too, in 2008, 2009. So there have been a few papers now.
This one has...
Is this
this is the case.
Is this... Yeah, so this is perfect.
This case is in the literature, and no one got it.
And no one got it.
Yeah, 45-year-old man, his 45-year-old wife, and 2 daughters.
Oh, so this is perfect.
And the sister-in-law.
Yeah. All right,
and
this is it.
Strongyloides.
People could have just Googled.
They could have Googled. Holy cow. And, I was Googling eosinophilia, fish, parasite, and wasn't getting Dientamoeba. But as soon as you said a family cluster. Bingo. That's it. Oh my gosh. This has got to go in the show notes, obviously.
Okay, great.
Folks, you missed an easy one.
I don't know if it was an easy one.
Professor
I think
Google.
It's how to properly Google to get the right answer.
Yeah, you have to do the right terminology, right? But there you go. Dientamoeba fragilis. And
so
now,
they had the idea that Baby the fish somehow had been contaminated, and that's how they had gotten this exposure. Now, the one thing I will say is one of the things they used to teach, and unfortunately some people still teach, is they say Dientamoeba fragilis, that there isn't a cyst stage, but they have actually now identified a cyst stage, which makes sense for the transmission of this.
now this is not acquired from fish, correct?
Somehow you're getting exposed to probably cysts, we now think, or the trophozoite stage. And that may have been on the fish. I mean, in these clusters, there was sort of the thought that maybe that was the common exposure, and that's how it got introduced. But it's not really, it's not a fish tapeworm. It's not a normal parasite of fish that we think about.
Yeah. So we're thinking of fecal contamination here.
It's probably. And then, the fish is kind of wet and nasty, and somebody has it, and then they're handling the fish, and then you eat raw fish, and yeah.
All right, so maybe the father had not good hand hygiene, and he prepared fish. He cut it up.
I'm blaming the sister-in-law, man. It's never the dad's fault.
And that's how they. Oh yeah, that's good.
She came and visited.
Fecal oral transmission. It's a trichomonad. It's an amoeba. Wow.
Yeah.
And this is also found in pigs and gorillas. So if you stay away from them, but unfortunately, humans is the problem.
Yes.
Traveler's diarrhea. Chronic diarrhea, fatigue, and in children, failure to thrive. Wow. That's great. That's a good one.
Well, my hope is in the future, right? People will have our either hard copy or electronic 6th edition of Parasitic Diseases, and then they'll just, find eosinophilia, they'll look through, and they'll see what the differential needs to be. And
how can they get a copy of the 6th edition, by the way?
Well, if they can go now to parasiteswithoutborders.com. And on there we have available now a free download of the PDF version. And if people want a hard copy, they can actually, there's a link there. They can go ahead and they can order a hard copy as well.
Parasites Without Borders, all one word.com.
All one word.
Or .org, actually.
Yep, but the PDF is free.
Yeah, it's great. So there you go, folks. Get your PDF of, it's all the latest stuff. Do you have this Dientamoeba in the book?
We do, actually.
You do.
There's, I actually quote Elaine Chiang's paper. No, actually, what it was funny, one of the chapters have a colleague, Craig Bootless, who is a physician up in Darwin, Australia, who I actually consulted when I was in Cambodia recently. We were seeing a lot of milioidosis cases and, asking him how he treats things. And he was one of the physician experts that reviewed our textbook. And he was the one who gave me a hard time that it took me a little while before I was willing to accept this as a pathogen. I'm a little cynical. I need to see a few cases. I need to know that it really,
So now everyone who wants to solve a case should get this book.
They get the book. They can, it's the nice thing, right? The PDF is you can search it. You can, take the clues, pop them in there, read the clinical presentations of different parasitic diseases, what the treatments, how to make the diagnosis. So,
And I'll remind listeners that this 6th edition, Daniel is now an author, so he's the one who's doing the case reports. So you can imagine that some of that leaked into the textbook since the last edition.
Yeah. And so for Michelle, I think we're still left with that challenge. Now we've got the information out there. We've got the textbook, hopefully accessible to a large audience. But how do we get them to read it? So that will be the challenge.
Dientamoeba fragilis. Oh, that's great. That's a good one. I'm still killed by the fact that it's so easily Googleable. It's just
exactly.
a killer. This episode is brought to you by CuriosityStream, the world's first ad-free nonfiction streaming service with over 1,500 titles and 600 hours of content. Founded by John Hendricks of Discovery Channel. And it was pointed out to me by Alan Dove on TWiP. He's no longer with Discovery Channel, of course. He helped found it, but he's now at CuriosityStream. And so knowing he came from Discovery Channel, of course, you will guess, and you would be right, that it's nonfiction. And you can get a lot of science here. You can see it on the web. You can just go to the website, curiositystream.com, and watch the movies there. Or you can use any of these devices that hook up the internet to your TV, like Apple TV, Amazon Fire, or Kindle, Android, iOS, Chromecast, etc. It's available in 196 countries. And what you get is a wide variety of science, technology, nature, history, interviews, lectures, for example. Stephen Hawking's Favorite Places is a brand new exclusive documentary where Hawking pilots a fantastical CGI spaceship across the universe, making stops at Saturn and many of his favorite destinations. I think you should do one of those for me, and I'll stop at many of my favorite viruses. How about that?
That's.
I went on a ride years ago at Disney World where the idea was that you were injected into the bloodstream.
Okay.
Did you ever go on that one?
I believe I did, actually.
And it's calm at first, and then something happens. I forgot what. You have to rush.
Yeah, there's like alert and flashing lights.
Yeah, but you sit in this thing and it moves. I hate that. I can't take that. You feel like you're floating, and I just get nauseous. But it's an interesting idea.
It's a little better than, what is it, the trip to Mars? There's some trip to Mars where you get so many G's you lose consciousness.
Yeah, they have another one where you're in the spaceship, and they had to add a lower intensity version because too many people were throwing up. But that's actually a cool one. I like that. Back to CuriosityStream, Digits, a 3-part series hosted by Derek Mueller. He created Veritasium, which is a YouTube science channel. Deep Time History, the story of the universe's 14 billion year history. So many cool programs. If you just go to the homepage, curiositystream.com, and search for science, you don't even have to log in, and you can just search. You get tons of cool stuff that you're certain to find interesting, as well as other topics as well. If you like history, you can find a ton there. They also are building their ultra-high-definition 4K library. They have over 50 hours of content. And the way you can see all of this material, you can get a monthly or an annual plan. You save a lot of money on an annual plan, of course. They start at $2.99 a month, less than a cup of coffee. Check out curiositystream.com/microbe. Use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 entire months free of one of the largest nonfiction 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign up. We thank CuriosityStream for their support of TWiP. Now we have a kind of paper that bridges TWiV and TWiP, I would say.
It does.
It's too bad that Dixon isn't here since he's on both podcasts. Paper recently published in PLOS Pathogens. It's an article called HIV-1 Integrates Widely Throughout the Genome of the Human Blood Fluke, Schistosoma mansoni. The first author is Sutas Sutiprapa, and the last author is Paul Brindley, from a variety of places. George Washington University, Mahidol University in Bangkok. You ever been there?
I was actually there in August.
In August?
Yes, very recently. Mahidol. It's actually an outstanding place.
Konkan University in Thailand, Wellcome Trust in the UK, Academia Sinica Taipei, Veterinary Research Institute in China, and the George Mason University. Did I say that already? I might have. Anyway, the problem here is that schistosomes are difficult to grow, and if you can't grow something, you're not going to be able to do genetics. And of course, genetics is the key to understanding how organisms work. Not too long ago, we talked about that CRISPR screen in Toxoplasma,
yes,
right?
Yes.
So Toxoplasma, you can grow them in cells in culture, so you can knock out genes individually and ask, what are they doing for the parasite? And you can't do that with schistosomes because they're hard to grow. And so what they did in this paper was to ask, could we infect them with a retrovirus? And if we could, then we could deliver genes or knock out genes and maybe do some genetics. So clearly the title of this caught me, and then I looked into it a bit, and I'm always skeptical. But it turns out that the data look really good. And nowadays, the extent to which you can do sequencing really helps you validate what you think is going on. In the old days, if you did this kind of experiment, didn't have any sequence, people would be skeptical.
Yeah, you'd be kind of lost. No, I mean, I was, you, as they say, you had me at the title with this one. You had both. You had HIV-1 and you had a parasite in the same title.
Amazing.
I mean, that's just what could be better.
Daniel, remind us what schistosomes cause. We've had some cases with schistosomes.
We
right?
have actually.
Of course.
So the schistosomiasis, and this is, I think we've talked about where you're in fresh water, and the infective form is going to penetrate your skin.
The cercaria.
The cercaria are going to penetrate your skin. There's going to be a snail intermediate in as we say, in all the ones that infect humans. There's a few exceptions in this group of parasites, but not that infect humans. So in humans, there's the snail intermediate. You're in the fresh water. And actually, when you get out of the water, it's not necessarily while you're swimming. When you get out and the water droplet starts to dry, that triggers the invasion process. So
they look for a hair follicle, right?
Exactly. And so what, when I swim in freshwater in Africa, I always immediately towel off, get all those droplets off before my skin can be penetrated. Dixon cringes when I tell him that's my approach to
he doesn't want you to swim at all, right?
Swim at all. I'm actually more worried about the crocodiles, but this was the classic clinical manifestations. People love the story when Napoleon's men went to Egypt, invaded, and they there's a story, I don't know how true this is, but they started peeing blood. There was blood in their urine. So one of the schistosomas, Schistosoma hematobium, will actually go to the venous plexus around the bladder.
Right.
And you can end up with hematuria.
Right.
The mansoni is the classic one for going to the mesenteric vessels in your abdomen. And the chronically infected children can end up with really significant enlargement of the liver and spleen, hepatosplenomegaly, these distended abdomens.
Right.
The japonicum, which is the one that had been native to Japan, was actually wiped out. It's the one that has, I'll say, maybe the highest propensity of the eggs actually metastasizing, spreading to the central nervous system.
Right.
And paralysis. The others can do this too, but so these are the different manifestations.
So this is a substantial pathogen, and we'd like to know more about it. So hence this paper. And some previous work that's been done, it's known that you can actually infect schistosomes with another retrovirus called murine leukemia virus, which the Goff lab, of which
the
Daniel
Maloney murine
studied for years.
Virus. Yes.
And the key to doing this is to substitute the glycoprotein of the retrovirus with that of vesicular stomatitis virus.
Are our listeners going to know what you're talking about?
We'll have to explain it to them. Now, vesicular stomatitis virus is a rhabdovirus in the same family as rabies virus, right? Bullet-shaped virus particles. It does not cause extensive human disease. It's really an aggregate. It's a cow pathogen, a pathogen of other rodents probably, and it's been used as a model virus in the laboratory for decades because it's relatively safe to work with. Although if you inject it into your finger, you will get a little rash on your finger. People have done that, dripping gradients to purify the virus, accidentally inoculating themselves, and they end up with a little bit of a rash. But it's relatively safe and easy to genetically manipulate. And they can take the gene. This virus has a glycoprotein in its envelope, just like a retrovirus does. They can take that glycoprotein and you substitute it for the glycoprotein of HIV or murine leukemia virus. This is called pseudotyping. So in other words, you now have murine leukemia virus, instead of its own glycoprotein, you have the glycoprotein of vesicular stomatitis virus. Now, the key here is that the glycoprotein is what is used to attach to the cell. It's not likely that schistosomes are going to have the same receptors for viruses as humans do, but it turns out that vesicular stomatitis virus is is able to infect an incredibly broad range of cells. Its receptor is low-density lipoprotein
maybe
related. Yeah, maybe. I know.
There was a paper. It was Mystery Solved, where
I know.
I'm not sure.
Not sure either. But whatever the receptor is, it's pretty broadly distributed because, in fact, people often use this glycoprotein to make viruses that can infect many cells. So you may be asking, well, if they already showed that murine leukemia virus with the VSV glycoprotein, we'll call those MLV VSV pseudotypes, can infect schistosomes. Well, why not use those for your work? Well, it turns out that MLV can only infect dividing cells.
Which is key. That's a key issue.
Yeah, because the nuclear membrane breaks down, and apparently that's when this retrovirus can get in, because retroviruses, of course, make a DNA copy of their RNA genome, which then goes into the nucleus and it's integrated into the cell. HIV, on the other hand, the lentivirus, can infect both dividing and non-dividing cells. And that's why it's used extensively for gene therapy applications, not licensed yet, but they're in development because, cells like neurons, which are never going to divide, you could, in theory, deliver something to them using a lentiviral vector.
Or stem cells, which I think
stem cells is the big one.
Therapy.
That's right. So they wanted to try and get these cells, these schistosome cells, infected with VSV pseudotyped HIV. So you take out the glycoprotein of HIV, which normally attaches to CD4. That's the receptor for HIV. But schistosomes don't have CD4, I'm pretty sure.
I do not think they do. I feel
so there would be no way that HIV would infect them, but you put on the VSV glycoprotein and you might have a chance. So that's the experiment. And basically they take the form of the schistosome. After you mentioned the what are the forms that are released from the snails again? I told you the name, and now I've forgotten what they.
So there's the
cercaria
and the metacercaria.
So when they get into you, then they transform into a schistosomulid.
Schistosomulum.
Right, and that's the
this
form.
Terminology is not easy.
That's the form they got a bunch of and can infect with HIV. The schistosomulum, schistosomules. Right, schistosomules. It's too hard. They add this VSVG pseudotyped HIV when they can actually see it sitting on the surface of the schistosomules by staining with antibody. So it looks like it's attaching to them. And then they can actually see that the virus gets inside and starts to undergo reverse transcription. The virus particle has within it reverse transcriptase, the enzyme that will convert the viral RNA into DNA, and they actually can see very shortly after infection synthesis of viral DNA. And they have something here called strong stop DNA. I have to tell you, because when I was a postdoc, Steve Goff was in the same lab that I was in, the Baltimore.
So you have to know what strong stop is.
They had just discovered it.
Oh, that's exciting.
And they were running these DELs with this tiny piece of DNA, because what happens is the RNA of a retrovirus is long, but the reverse transcription begins very near the 5' end because there's a tRNA primer there. And it just makes a short piece of DNA, and they called it strong stop because when they ran a gel out, they get a big smear of products, but there was this big band of really prominent, and it was short, so they called it strong stop.
I have to admit, I think that's fascinating, the whole idea that it primes, that it starts the transcription with a tRNA, and then it forms this little basic, which is going to be a primer to then
goes to the other
end where there's the homology, and then you're going to get,
because the ends are repeated.
I would tell our listeners that if you do anything in HIV, this is my equivalent to the immunologist who you have to mention the surrogate light chain to show that you're a member of the club. Well, the strong stop.
That's right.
Now you're a member of the HIV club, I think.
They also look at the full length plus strand DNA, which eventually gets made. But it's interesting that this works. Because, in fact, if you heat and activate the virus particles, you don't get this DNA made.
Yep. So you don't see the strong stop, and you don't see the positive strand HIV cDNA.
I'm already blown away that the RT will work, because we know that there are probably some cellular factors involved, and apparently they're there. And
yeah, I wonder. I mean, if you go back right to the original work where they describe RT, I mean, all they did was a little bit of detergent, and then they throw in the ACGT, and the RT is sitting there waiting to go, and as soon as it gets the building blocks, it puts them together and makes it so.
So maybe RT is not so surprising.
RT, I was like, okay, that seems fine. You've got ACGs and Ts, and you're going to go ahead and you're going to make your copies. Because you're going to start with your RNA, then the virus. You're going to go ahead and make your DNA copies.
The real question is whether it integrates right into.
Yeah, I mean that was my thought. Okay, so that's fine.
And it probably wouldn't, right?
Well, you would think is it going to somehow it's going to be able to get into the nucleus,
right?
Because we think of two things in I'll say the human and the primate system is that there's cofactors that apparently help it to get in, actually help it to bind and integrate,
right?
And there's even restriction things. So the restriction might not be there, but would the compatibility with the cofactors be there in a schistosomulum, in a schistosome? I mean,
it's apparently yes, because they then extract DNA from these schistosomula. They do PCR, and the PCR strategy is kind of cute. They use a primer in the GAG gene, one of the genes of HIV, and then a primer from the genome of the schistosome from an endogenous retrovirus-like element.
Yeah, these it's amazing. Yeah.
So these organisms probably were infected or exposed to retrovirus-like nucleic acids many, many years ago, and they're still stuck there in the genome. Well, the bottom line is they can see that the HIV genome is integrated into the schistosome.
That was shocking. It can get into the nucleus in non-dividing schistosome cells. They're not dividing, and it can integrate.
No, it is amazing.
That's pretty amazing.
Now, here's the part that is a little weird. This is a good experiment. They use a couple of inhibitors, which are antivirals. They use azithromycin, AZT, the very first HIV antiviral, right?
AZT.
And the nevirapine, which are two RT reverse transcriptase inhibitors. They use a integrase inhibitor, but none of them block.
Yeah, I have to admit that made me think. That sort of, made me pause.
Gave you pause. But they did simply add these drugs to the schistosomula, so maybe they don't get into schistosomula. Right?
Yeah, that was the only explanation that made sense for me, is that they were not penetrating.
But
they were not getting in.
For me, it's a key thing. I don't know. I mean,
it really is key because they do have heat inactivated as our control.
Yeah, that's fine.
I usually use nevirapine. Actually, is a lot of one of my negative controls. I'll heat inactivate virus. I'll use nevirapine. So when the nevirapine didn't impact it, that sort of made me a little concerned. But they do have heat inactivated, so they do have a negative control.
That's fine.
I would have liked these negative controls also.
Yeah, with heat inactivation, they see very, very low integration, which is probably just background. So that's good, but I wonder if they could have tried to permeabilize the schistosomula in some way to get the drug in, just to show that it would work.
Because it did, and because I'm sure the reviewers were asking them what's going on.
I mean, the data we're going to give them now is really incontrovertible, right?
Yeah, I mean,
it's clear.
Yeah, we've got the sequencing. We see it integrating. We've got the exact sequence of the integration sites. We can count integration per.
Yeah, it's quite clear that it's happened. It's just puzzling, and I suppose the non-permeability just explains. That's the kind of experiment you'd like to have, but given what they show us later, you don't need it.
Yeah, you're okay with that.
You're okay.
That's why it still got published, because people said, you know what, it's still...
Yeah. So they now know that the HIV is integrated into the schistosome, so they want to know how many times and where. So they sequence first they sequence the PCR product, right? And they find lots of integrations, 8,000 different integrations, all throughout the 8 chromosomes of the parasite, 7 autosomal and one sex chromosome, right?
Yeah. So these, if our listeners may remember, this is the wonderful story that people like to tell, where the male schistosome wraps himself around the female schistosome, and they actually they say they're in copula. They're actually constantly having whatever sexual exchange going on, and it's this like sort of the swans of the parasitic world are mating for life, and with some exceptions. I think every so often the women leave and find whatever schistosome happens to be around. But
they also find integrations into mitochondrial DNA. That's interesting, which we don't see in mammalian cells, as far as
that
I
is
know.
interesting.
Can't get in there for some reason. Now, this is something that was really an important issue a long time ago when we were trying to figure out if the retrovirus XMRV was responsible for a human disease known as chronic fatigue syndrome. A number of people were said, "Oh, look, the virus is integrated," and one of the key pieces of evidence of retroviral integration is that you get dinucleotides at either side of the integrated genome, and you get duplication of host DNA. That's the mechanism of the integration reaction. They can see the diagnostic nucleotides at both ends of the integrated HIV and the junctions that are duplicated. So that's quite clear. They also then sequence the entire genome, not just the PCR product, but the whole genome is sequenced, so they can get the number of integrations per worm, right? Because the previous one you're amplifying, so you don't know actually how many integrations there are. And what they did was they used 5,000 schistosomula that were infected for this experiment, and they get 2.1 integrations per schistosomulum, and each schistosome, 2,100,
yeah,
2.1 integrations per nucleus. Because of
because there's about 1,000 nuclei in a schistosomulum.
Right. It's not just one cell.
It's not just one, and I think that's important. It's not just one cell. This is an average of 1,000 cell, 1,000 nucleus.
Right. So 2.1 integrations per nucleus.
Yes.
Which is good if you want to, and that's an average, of course. Some are going to have less and some are going to have more. But that's a good number if you want to start disrupting genes, right? Just one gene per nucleus. That would be.
Ideally, that's what you want. Ideally, you want one. Let's say this has a siRNA in there, or a short hairpin RNA, or a CRISPR cassette, or whatever you want to put in.
Right.
Ideally, you want to put in a specific thing. So you can adjust your titration, how much virus you expose it to.
Right. Now that's what they want to use this for. They want to deliver genes or disrupt genes. And I thought there was one more experiment that they could have included that's just screaming in its absence. Is there any one thing that you would have liked to see? There's one thing for sure, and that is they never ask if the integrated provirus is transcriptionally active. Is it making RNA? A piece of cake experiment, right? Because if it's silenced in all integrations, that would be really interesting. And of course, it would be a problem for what they want to do. They want to use this to make like Cas9 or whatever.
Right? Yeah. It's interesting, right? So they use some terminology. They talk about they're using this PNL 4.3 vector that they're then pseudotyping to do this. Now, when you
have, it's right here.
I have.
It's everywhere.
It's everywhere. It's everywhere. When you buy it commercially, it has a luciferase in there. And then we do things where we'll chop out the luciferase, we'll put in a GFP or a red fluorescent protein or whatever. So it's interesting that, it seems like they probably could ask this question.
Piece of cake.
Yeah.
Especially if they have luciferase
unless they've done something where they said, we don't want the luciferase, but if they took out luciferase, they would put in a fluorescent. So I see what you're saying.
It's funny that it's not there.
It is funny that they didn't go to the next step.
It's not key for the paper, right? Because they're looking at just integration, and maybe they're saving it for their next paper,
Maybe. What I was excited or would like to see next is to have this in a germ line, so that you're actually creating, sort of knockout schistosome lines.
So what can you do with a schistosomulum in culture? Nothing? It just exists for a while and then it dies, or do they grow? See, this is where Dixon might have been useful, right?
Exactly.
No, he's useful for many things.
No, this would have been a perfect issue, because that's kind of the question: is how do you go ahead and use this system now that you could do it, now that you can jump in, can you somehow look at penetration or different things, and that I'm not sure about.
Yeah, it's not clear. establishment of transgenic lines. So they do want to make lines, which implies that, some derivative of Schistosoma can propagate in culture. But I'm not familiar with it. I'm sure Dixon might know, or a listener might know as well.
Actually, that would be great.
Did you run this by Steve Goff at all?
I didn't, actually.
I'm wondering what he thinks of it.
I mean, I wonder about, so as they mentioned, people have done similar things with like strongyloides, for instance, where they actually go after the germline. Again, where's Dixon? Could you use this in trichinella? If this can get into to this schistosomulum, could you pop this into these timed larval stages of trichinella? And then, Dixon's all, has all this whole interest in the, was it the stichocytes? Do you start knocking out different genes in the stichocytes and seeing, what the impact would be? So I actually really like the idea of this as a tool.
So let's see if Dixon answers our question.
Because he can write in. He's in Bolivia.
The next time next time he's on TWiP, we'll know if he listened or not.
Exactly.
Because Dixon, we're asking you a question and several questions, and we'd like an answer. And if he doesn't answer us, it means he never listens.
Yeah. Well, I think it means he didn't listen to this one.
I think I would.
We'll extrapolate. Do we extrapolate? Okay.
For sure.
Okay.
And remember, everyone, parasiteswithoutborders.com. You can get your free copy of 6th edition of Parasitic Diseases.
Yeah.
Parasitic
diseases.
And then you can figure out all the case studies. Speaking of case studies, I bet you have another one.
No, I'm all out.
You're all out?
No.
You said it was not possible.
I have a stack. So let me pull one off the stack. All right. Now, this one, I feel like the last one was a little tough, and I think we saw that. We had a limited number of people writing in. So hopefully this one will be a kindler, gentler case study, and we'll see a lot of our regulars writing in as well as, I applaud the people that wrote in. I think those were really well thought out.
I wonder if people don't write in because they don't know the answer.
They don't know the answer.
But some people, none of these knew the answer, right? But they tried.
Yeah, they tried. And that's what I would say. Try.
Yeah, I would encourage folks to try, because when we only get 4, we think you don't like us anymore. Right?
Is that what you were thinking?
Yeah, they're getting tired. Whereas we know people like the case study, right?
Yeah.
So
I think of this as a safe place. We're all here to learn. As I pointed out, when I initially heard this case, even when they were reading off, I still wasn't sure. It actually took me a little while after the fact to read the literature and convince myself. I should have just known ahead of time, because Elaine is about 3 times as smart as I am. So if she thinks it was Dianthomia fragilis,
then it was,
it
right?
is.
Okay.
So
a kindler and gentler case.
Kindler. This is welcome back to Thailand, but we could be in Peru, we could be in Ecuador. This could occur in several places. But this is a 25-year-old Thai woman, and she comes into the hospital clinic to be seen with a chief complaint of facial swelling. She reports that she eats a typical Thai diet, pickled fish dishes, all the other things that we've talked about that I enjoy eating as well.
What was that, jumping salad?
The jumping salad, which I guess is popular in the Philippines and in Thailand. What is it? Sumtam. So there's a lot of crustaceans and fish and things that are just sort of cured in lime juice and other things, not necessarily cooked. And she now has this facial swelling, and it's migratory. It's not just one area. It appears to be moving. She says that the swelling is not tender, but there's a mild itchiness to it. She's had this for about the last week, and she doesn't report any pain. The only thing is this just mild itchiness, and the fact that it's on her face, and the swelling is in different places, different locations. It seems to be moving.
Migrates around her face.
Seems to be migrating.
Not off of the face, though.
Not off of the face.
All right.
And she's healthy, no past medical history, no past surgical history. She's not allergic to any medications that she's aware of. Family history, she says everyone's fine, no significant family.
She's married, she has kids?
She is married and she does have kids.
She's HIV negative, I presume.
She's HIV
no drug problems or anything like that, right?
Nope, no drug problems.
And she has not traveled, right?
No, she lives in Bangkok and has been in sort of the immediate environs.
What does she do for a living?
We don't know, actually. She doesn't tell us this. So
not voluntarily, but
she doesn't voluntarily. She doesn't tell us.
Okay.
She's asked, but just,
and her family's okay, right?
And her family, she's the only one with this problem.
And how long she had this? About a week,
It's been about a week.
And does it correspond with anything different she did with her life?
No. It's just out of the blue. She's been doing her normal, usual stuff.
Okay.
And now she's got this swelling, which seems to be migrating around her face. And when we examine her, she actually has this swelling, which is on the right side of her face. But she says it hasn't always been, as mentioned right there. It's been in other areas. So that's just where it is when we see her.
How much of a swelling is this?
It's actually pretty significant. You can see it from several feet away. It's I would say probably about 3 or 4 centimeters raised. A little bit of redness, not very red, but there is some say erythema, a little bit of increased redness.
And so it's mainly fluid there that's causing this?
That's interesting. So if you feel the area,
yeah,
it's firm. You don't feel, say, a fluctuance.
Okay.
It doesn't feel like there's a pocket of or an abscess or anything like that.
So it's cells that have come in.
It's swelling. Yeah, it's actually sort of edema in the region, consistent with edema, where it's just firm.
It's all right. So by the firmness, you can't tell if it's cells or fluid or both, right?
What I would say is it doesn't feel like there's a fluid collection. It doesn't feel like there's a pocket of fluid, which we would refer to as fluctuance. It doesn't feel like there's an abscess that needs to be drained.
Okay. And she doesn't have a fever, right?
No fever.
And her GI tract is fine?
Yeah. No. That's interesting. So right now she doesn't have any diarrhea. And that's one of those tough questions because we do ask, like, have you had any issues with diarrhea maybe in the last month or so? And there was some, I did have some loose stools, but I get loose stools every so often.
Yeah,
so it's sort of, we're not sure at this point whether or not that is associated with this, but she did have some loose stools prior to this, but that's not uncommon.
Mm-hmm. I was going to ask you something else. Now I've forgotten what it was. GI tract is okay. Eosinophilia.
So we don't have any blood work yet.
Oh yeah, you're going to tell us that next week, right? Next time.
See if I tell you that next week. Maybe I'll tell you that next week.
Okay, that's fine.
But I'm not telling you today.
Yeah, right. Got it. Facial swelling and unexamined.
Should I tell you? Do you think we need that? I'll give that to people. The white count is up, and there are eosinophils.
Okay.
Just we'll throw that because I'm being nice.
There's eosinophilia.
There is eosinophilia.
Okay.
So elevated white count, increased level of eosinophils.
Got it.
And this migratory facial swelling in a woman that likes to eat fish that is not always cooked.
Is that an important part of this?
And there may have been some loose stools previous to this, but we don't know if it corresponds.
Fish that is not cooked. And is that important, do you think, for this? You can't tell us.
I can't tell you.
And she lives where in Thailand? In a city or a rural setting?
She lives in Bangkok, in the city.
Bangkok.
In Bangkok. Which is really an amazing city. I have to, it's, people describe Venice with all the water and rivers everywhere. And Bangkok is, it's right on this really large river. And then there's all these small rivers. And people have these homes built right in the rivers, some of them up on sort of these stilts. And, people are swimming and washing their clothes. And the boats are going by in the rivers. It's a beautiful city.
All right. And the facial swelling, which is firm.
So
when you push it, it doesn't leave an indentation, right?
True. It doesn't.
It's firm.
Non-pitting edema.
And it moves around.
And it seems to be moving, and doesn't seem to be an abscess.
All right. I think that's enough. It's enough for me.
I think people should be able to get this.
Should be able to get this one. We should have at least 10 responses for this one.
Yes.
All right.
I think so.
And you know what? The winner will get a PDF of parasitic disease.
Actually, yes. Why don't we do that? And if you, even if you just email in, you could have a free copy.
That's right.
Of the PDF.
And now people will say that we're being mean, but we're not. We just like we like when you write. I want to tell you about a sponsor of this episode, Drobo. makers of expandable and safe and simple storage arrays for your computer. They connect to your Mac or your PC. They are arrays of hard drives. You can get 5 or 8 or 12 drive bays. And they're very different compared with other kinds of drive arrays because they're designed to protect your data forever and to be very simple to use. And as I've told you before, the whole company was started by a man who lost his honeymoon pictures because he had them stored on a RAID system, and he couldn't recover them. And so he developed Drobo, which is different from RAID. It's beyond RAID. It's a RAID-like technology, but it's a patented different RAID, which they call beyond RAID, because you protect it against 1 or 2 drive failures. If a drive in your array, let's say you have a 5-drive Drobo, and you've got your photographs on them and your movies and your data, if a drive fails, you're not going to lose any data. You can just take the drive out and put a new one in, and because of the nature of the beyond RAID, all the data will be reproduced onto that drive as well. So no loss of data. In fact, you can be protected against up to 2 drive failures. And there are all sorts of other protections as well. For example, let's say you're saving your valuable photos to the drive and there's a power failure. Well, you're not going to lose them because there's a battery-backed cache to store your data so that next time when you power it up, it'll be there. Now, you can buy Drobos, as I said, in different capacities. If you lose a drive, how do you know? Well, a red light goes on next to the drive. It means the drive doesn't work anymore. If the drive is, and then you just pull out the dead one, you put a new one in, bingo, you're ready to go. If you see a green light next to every drive, that means there's plenty of room and everything is working correctly. If you see an orange light, that means one of the drives is getting full, the one next to the orange light. And when it's red, solid red, then it's full. You need to replace it. You pull it out and put it in a bigger one. You can get up to 10 terabyte drives these days. Wow. So you could have a 500 terabyte Drobo if you buy the 5-bay one. 500 terabytes. What's the next one up from terabyte? Gigabyte, terabyte. Ooh.
I don't know. This is going to require some Googling.
Let's say 1,000 terabytes is a petabyte. A petabyte.
Petabyte. Okay. That's a lot of storage.
That is a lot of storage.
You know, because Dixon isn't here, I was thinking about. So I always claim to my children that I invented the hard drive. I've probably never shared this story, but you're talking about the hard drive. Now, I was back in high school, and to date myself, it was the early '80s, and I had one of these IBM PC Junior computers.
Yes.
And there was
They didn't ship many of those.
No, it was special. It did not go well.
They canned it after a while, yeah.
It didn't go well. But they had the real floppy drives, right? They're 5 inches or so, and they were really floppy. And there was a word processing program called WordStar, and you'd put the disk in,
right.
And then when it was time to save, it would alert you, and you had to stop, not touch the keyboard, pull out the disk with the software, put in the disk to save. So I grew tired. I'm lazy.
Horrible.
So what I did back in the early 80s is I actually hardwired in a second hard drive. And I left WordStar.
Floppy drive.
Well, it was actually a floppy drive. It was a second floppy drive. I left the WordStar in there. And then what had been the primary drive was now where it would automatically save. And I predicted the fact that, you know what? You could just, for each different program, you could have another floppy drive wired in, and you wouldn't have to keep pulling the disks in and out like that.
In and out was ridiculous.
And
I used to write grant applications on those big floppy drive-based computers, pre-Mac. And I remember going back and forth and back, and then it said, sorry, there was an error saving your file.
Exactly.
Oh, thank you. Thank you. What do I do now? No idea.
Nothing.
No idea.
You can't go back. You can't.
You're toast. I got rid of those, boy.
So look how far we've come.
We have come. And now we have Drobos where you're protected forever. It's simple to use. It's all based on lights. All you have to do is fill them up with hard drives of your size.
Now, but they're green and red. I feel bad for colorblind people out there. There needs to be some, why green and red? Why the two colors that are so hard to see?
They like to say if you can read a traffic light, you can...
you can use a Drobo.
But you're right. What fraction of the population is green-red colorblind,
I think it's like 1 or 2% of men, right? Do we know?
That's a good question. I guess Drobo has made the decision that, I don't know.
I wonder if it varies in parts of the world as well. Let's see. So it says color blindness affects, they say 1 in 12 men, 8%. Doesn't that seem high?
Seems,
but then they say
high.
In Britain, this means that apparently it's less common in women. So 4.5% of everyone in England.
Anyway, Drobo thinks you will gladly let go of all the knobs and levers other RAID systems force upon you for the simplicity of knowing your data are safe and that you can sleep at night. Microbe TV listeners can save $100 off of their purchase of a Drobo 5D, 5DT, 5N, or any 8 or 12 drive system. Just go to drobostore.com and use the discount code microbe100.
And for the 4.5% that are colorblind, just have your friends stop by periodically and
That's
let
what
you
I was
know.
thinking.
Let you know.
They'll say, hey, my partner, is that green, that one, or is it red? It's blinking. Well, if it's blinking, they know it's got to be red blinking, because it doesn't blink at any other color. We thank Drobo for their support of TWiP. In fact, one of the workers at Drobo, Mark Fuccio, who we've known for a number of years, who helps get us these sponsorships, he was on TWiV on Friday. That's TWiV 412. Is that it? How can I not know what episode TWiV is at? Let me look it up, because he had interesting things to say about Drobo. 412. So take a listen to that. We have a few email here to round up this episode of TWiP. The first is from Sophie, who writes, Dear TWiP team, thank you for the story about schistosomiasis and how they eradicated it in Japan. I found it very interesting. To answer your question, Dixon, I graduated med school in 2011 from Aarhus University Faculty of Health. Unfortunately, I don't remember who of my microbiology teachers revealed this story about Kadayama fever and schistosomiasis in Japan, but obviously it made an impression. Have a very nice day, and thanks again for expanding my horizons. And of course, Aarhus University is not in Japan, right? It's in Denmark.
Denmark, yep.
Mm-hmm. Why don't you take the next one, Daniel?
Derek writes, Hi, there's a paper out in Nature Communications that would be appropriate for either TWiV or TWiP, but probably most so for TWiP, Eukaryotic Association Module in phage WO genomes from Wolbachia. This is fascinating. As far as I know, the first time a phage has been shown to carry around animal DNA. In this case, the DNA for the black widow spider venom. Would be great to hear you take on this work. Also, when I went to your website to remind myself of your email address, the entire page was shortly taken over by an ad for a virus removal company that tried very hard to raise pop-ups, preventing me from closing the page. I don't know if your website is a WordPress site, but it is very reminiscent of an attack on a site I maintained that happened a year or so ago. Keep up the great podcast. Thanks, Derek.
Well, Derek, we did that paper on TWiV this past Friday, TWiV 412. It's a very cool paper. Now, you thought it would be good on TWiP, probably because Wolbachia is an endosymbiont of eukaryotic cells. But we decided that it was a virus story, so we did it on TWiV. I don't know anything about this this ad. It certainly has nothing to do with I do have a WordPress install for the microbe.tv sites, but no one else has complained to that, and I'm not aware of how a virus removal company would do that. It's probably independent of my code, I would guess. I don't know. If anyone knows more, let me know. We have one from Anthony, who writes, back in the recession of the early 90s, the tide of yuppies on the Jersey City waterfront for a while reversed. Left behind were here and there some yesies, young educated scroungers, myself among them. Not far away from where I lived on Warren Street by Sussex, there was a block square ditch from construction suspended due to the inclement economy, the Onyx site. The fallow land had become marsh. After the spring rains, each night from the muddy water came clouds of mosquitoes and the unbelievably loud chorus of mobs of toads mating. Sleep was difficult, for even with screens, mosquitoes found their way into the apartment. Plus, even a block away and 4 stories up, the noise from the amphibians kept me awake. The thought came to me while staring at the ceiling one night that the biblical plague of frogs must have been malaria. The Egyptians associated the unusual sound with the disease. What I couldn't understand was why they didn't blame the mosquitoes. Professor Despommier seems to have answered that when discussing the culturing of mosquitoes. He mentioned that the volunteer feeders became desensitized to the bites. Look at that. Dixon's not even here.
He's getting all this credit.
And he answered the question. And now another topic. In a discussion of toxoplasma, there was some wondering on what attraction cat feces held for mice. Many years ago, when I'd go walking in the woods, I'd ever so often see animal skulls. I never saw a carcass or a skeleton. My surmise was that when an animal died, scavengers made quick work of everything, including the bones. The skull seemingly was too hard for small teeth to dent, and so was the only thing that remained. Domestic cats tend to catch small rodents and birds. The felines consume these by using their jaws to crunch the body and so break the bones. Then they swallow everything in chunks, leaving just the head or wings. The undigested bones are passed. Perhaps the calcium source is what interests the mice?
I like that idea.
The calcium source is what interests the cats, no?
No, I'm thinking so the mice would eat the cat feces.
Oh, I see.
Because it would have the bones
of course.
in there. Yeah.
I don't know, Dixon,
I know
what do you think?
I know, out west, it's actually fun. We were watching a little documentary on Yellowstone, and then you see these two little kids in these brightly colored jackets run through. It coincidentally was our kids. But there are these arches at the at this park in the center of the town of Yellowstone, and it's the antlers. And a lot of the small animals, one of the things they have trouble getting in their diet is calcium. So they gnaw on the antlers. So there is actually a challenge for a lot of rodents to get an adequate amount of calcium, and I like this.
But dogs also eat feces, right?
That's just gross.
Yeah, it is gross, but eat it.
What is it? Coprophagia? Coprophagia.
Yes.
Yes, I discourage that from.
Well, Dixon, you should read these. You should listen to these letters, Dixon, because they're very interesting and relevant to you.
Yes, you would. Dixon would like these.
He would. Why don't you take that last one?
Bryce writes, TWiP triad. This includes you, Dixon. This case comes at a great time since I have a test this Friday about hemolysis and other blood disorders. Given the lower levels of hemoglobin and mean cellular volume, as well as the slightly elevated bilirubin levels, this woman is presenting microcytic anemia. Her symptoms appear to follow a cyclic fashion, which I think this is a guess for one of our cases, which combined with the anemia suggests Plasmodium to be the diagnosis. To parse out the specific species, I'm sure a PCR could be ordered. However, about following the nature of her fever, the patient appears to be infected with either P. vivax or P. ovale, as these parasites give a spike in fever corresponding with the rupture of the schizon, followed by a couple of days of rest while the parasite infects other RBCs to continue the cycle. P. falciparum, on the other hand, undergoes continuous schizant rupture following to a fever that never breaks, leading to a fever that never breaks. Additionally, these two species, P. vivax and P. ovale, are associated with an enlarged spleen. To treat the malaria, chloroquine to eliminate circulating parasites should be administered in addition to primaquine to remove the hypnozoite that can cause a relapse infection. Thank you for the almost weekly study material through the case studies. Hopefully I did not get this one wrong. Otherwise, that does not bode well for my test.
I think this was the woman from Guinea.
I think so. And it was malaria. It actually was falciparum. And there is a cyclic fever with falciparum. It often doesn't establish when we see it in a traveler because it takes a while for the synchrony to occur. I think it was Hippocrates, right, who described the different periodicity of fever. There can be a different characteristic periodicity to the fever, but we usually go ahead and look at the blood smears we did in this case to identify which type of Plasmodium.
Yes, Bryce's was just a bit late. Understandable, studying for a test. No problem. All right, that's TWiP 119. You can find it at iTunes, microbe.tv/TWiP. And of course, on your mobile phone, there are a whole host of programs, apps that you can use to listen to podcasts, and you can just search for TWiP on those, although you might get photography podcasts if you just search for TWiP, so just use parasitism.
This week in parasitism.
Week in parasitism. Also, think about becoming a patron of TWiP, and the Microbe TV family of science shows. You can contribute as little a month. You can go to patreon.com/microbetv. We have other options at microbe.tv/contribute, like a one-time payment or buying things at our Cafe Press store, or even buying things at Amazon. If you use our affiliate link, we get a percentage of the sales without any additional cost to you, and it goes towards our shows. Now, you may ask, why do we need contributions and funds? Well, we do have expenses associated with all these shows, but more importantly, we'd like to do more. We'd like to go places and do podcasts in your hometown. Do it in front of an audience.
Depends where you live.
Yeah, right. Lebanon, Oregon, sure, we'll go there, as long as it's near a major airport. And we need money to do that. And and also, I could use help, a little bit of part-time help running this enterprise. And that's why we want to raise a bit of money for that. And if you feel motivated, please do. If not, no problem. We understand it. That's why we do ads as well. Send us your questions, comments, and guesses for this week's case study. TWiP at microbe.tv. Of course, today, Dixon de Pommier is in Bolivia. And next time he shall be back. He has told us he'll be back. So if you missed him, don't worry, he'll be back next time.
It'll be okay.
Daniel Griffin is here at Columbia University Medical Center. Thank you, Daniel.
And I guess at parasiteswithoutborders.com.
Parasiteswithoutborders.com. It is a new website. And Dixon and Daniel and
Chuck
Knirsch are involved with the website. It is to distribute the book and get
more education about parasitic diseases out there to those that need it.
Right. So check it out. And as we said, you could download a copy of Parasitic Diseases, 6th edition. Spread the word.
And thank you. A pleasure as always, Vincent.
You're welcome. And in the beginning, we had no problem with introductions, did we?
It was very easy. You won't have to edit and do cutouts.
Nothing. I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiP is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. I'd like to thank the sponsors of this episode, CuriosityStream and Drobo. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.